So Chapter 5 starts to look at the structure and function of the large biological molecules, or the macromolecules. There is 4 main classes of macromolecules, so the name macro tells you that they are big molecules. You've got the carbohydrates, lipids, proteins, and nucleic acids. And their architecture or structure is going to explain how they work. When it's a polymer, it means it's a long molecule consisting of many similar or identical building blocks linked covalently. Legos would be a good example of something that's like a polymer. You've got lots of pieces that fit together and you just keep hooking them in a line on there. Monomers are going to be the repeating smaller units in there, the individual molecules that serve as the building block. So you could have each individual Lego is a monomer, but you hook them together to make a polymer in there. So there's some ways that this can happen. Sometimes when you do this, you'll have a dehydration reaction. And you got to let the names give you some hints in there. So when you dehydrate something, you're removing the water from it. And that's what you do in this type of reaction, is you've got a short polymer and an unlinked monomer here. You're going to pull a hydrogen off of one, a hydroxide off of the other. So you remove a water from the two, and that glues it together. The opposite is hydrolysis in there that's going to break down a polymer. That's going to be where you put water into it, and you can have the hydrogen from the water attached to the one, the hydroxide attached to the other. So the dehydration reactions are sometimes referred to as condensation reactions in there, because you're going to have the loss of water occur in there. So when water condenses out of the air, it's coming out of the air. When you have a condensation or a dehydration reaction, water is coming out of the reactants. A lot of what's going to control the rates of reactions are going to be enzymes in there. These are going to be macromolecules that will usually speed up or control reactions in cells. So hydrolysis, again, this is that breaking down. An example of that is digestion. So when you digest food, you add water into it, and it helps with your digestive processes. So the carbohydrates, these are going to include sugars and polymers of sugars. Sugars are also called saccharides, so mono is one, di means two, poly is many. Some of the sugars are aldoses, some of them are ketoses, based on where you have this double bond with the oxygen. So aldehyde on the end, ketone is in the middle in there. And everything else looks the same. One is glyceraldehyde, one is dihydroxyacetone. So any idea where you might find dihydroxyacetone? It's actually the stuff that's in self-tanner. Acetone is a nail polish remover, but dihydroxyacetone, when they talk about, oh, the self-tanner is just a sugar, yeah, it is a carbohydrate, but it's not sugar like table sugar like you're thinking in there. And it does have a distinct smell to it. So if you've ever used self-tanner, you notice that it has a smell you can't quite get rid of in that lotion. No matter what they do to it, there is a smell that haunts you with it. And this is why. So if you've got the 5 carbon sugars, you've got ribose and ribulose. The 6 carbon sugars, glucose, galactose, and fructose in there. So the monosaccharides are the single sugars. All sugars are a multiple of 1 carbon, 2 hydrogens, and 1 oxygen. So if you have a pentose, you multiply everything in there by 5. 5 carbons, 10 hydrogens, 5 oxygens. If you have a hexose, you multiply everything by 6. But that is the common multiple in there. Glucose is the most common sugar, and it's going to have a carbonyl group and multiple hydroxide groups on there. So your aldoses or ketoses are going to be, sugars are considered an aldose or a ketose, depending on where that double bond is. They get named by the size of the carbon skeleton, so hexose, pentose, triose, 3, 5, 6, and then the spatial arrangement of them in there. Your monosaccharides are your major nutrient that's used as energy for cells. So that is where your body is going to obtain energy. A lot of times people think, oh, I want proteins for energy. No, you don't. Carbohydrate is what your cells like to run on. Some things like your brain won't run on the other fuels unless they are absolutely forced to, and then they don't run well on it. So the disaccharide is going to be where you have two monosaccharides that get joined by a glycoside linkage in there. Basically, you have a dehydration reaction. So you would want to know what these disaccharides are. These are the really common things that you'll run into all the time. So maltose, sometimes called malt sugar in there, is made up of two glucoses. Lactose, your milk sugar, is a glucose and a galactose. And sucrose, which is table sugar, is going to be fructose and glucose. So polysaccharides are going to have multiple monosaccharides that are joined by those glycoside linkages. So a starch is going to be stored glucose in plants within the plastids. So they're going to be stored in plastids that include your chloroplasts in there. Glycogen is going to be the polymer that's stored in animals. So both plants and animals can store some sugar in there. We store it as glycogen, plants store it as starch. In the starch linkages in there, most of the glucose monomers are going to be joined by a 1,4 linkage. It means carbon 1 is attached to carbon 4. So example would be maltose in there, and they'll tend to end up being helical in there because of the angle of the bond. So amylose is a simple starch, it's unbranched. Amylopectin is a little bit more complicated. It's got 1/6 linkages in there, and it's branched, so it will have some little branches that form off of it. Some structural polysaccharides in there. Cellulose, this one is a tough component of cell walls. You've got the two ring forms of glucose in there that are going to be either alpha or beta in there. Starch is all alpha. Cellulose has some beta in there. It's straight and unbranched. And what's different about this is there's not very many organisms that can hydrolyze the beta linkages. When you have the alpha carbon attached on there, we've got enzymes that will break that down. We don't have enzymes that will break down the beta linkage. Can you think of something that people eat that probably has some beta linkages in it? Corn. We all know what happens to corn when it comes out the other end. The reason it still looks like corn is unless you've chewed it up and manually broke it up with your teeth well enough, you don't break up those beta linkages in there. And that's why it comes out that way. Same thing with grass. If you have a dog, a lot of dogs go out and eat grass. And out the other end of the dog comes grass. They eat long grass. Occasionally you get long grass out the other end. They can't break down the beta linkages in there. A cow, on the other hand, does have bacteria in its gut that help it break down those beta linkages. So you can feed the cow the corn and it's going to get completely digested in him. So insoluble fiber in your diet is going to be mainly cellulose. It's not meant to be broken down in there. It's meant to come out whole on the other end, and that fiber is going to help to absorb things and kind of scrub in the gut because it doesn't break down. Chitin is a polysaccharide in arthropod skeletons and a lot of fungi. So that's what is going to give bugs their hard shell on the outside in there. And that can be fairly hard if you've stepped on the large beetle or something where you can feel it crunch. Yep. So this is showing here where you've got starch with those linkages here. You notice they all look the same. Cellulose, you've got one that goes up, one that goes down. That's where you get the alpha and beta from in there. So your lipids, all lipids are hydrophobic. They don't want to mix with water in there. These are going to include wax, some pigments in there, fats, phospholipids, sterols. So fats are constructed of glycerol and fatty acids. So a fatty acid is a long chain of carbons. Usually in biology we're looking at 16 or 18 carbons in the chain. At the end you've got a carboxyl group that's going to give it that acid end on there. They're relatively nonpolar. So you've got this really long chain here that's all symmetrical and just a little bit that's polar at the end. That's not enough to consider it polar in there because it's just so much more of it is nonpolar in there. And that's why fats are not going to combine with water. Glycerol is going to be an alcohol. It's 3 carbons long and each is going to have a hydroxyl group on there. So when you make a triglycerol or triglyceride, what you do is you put 3 fatty acids onto a glycerol in there. And the reason that's significant is most stored fat and most consumed fat in humans is in the form of a triglyceride. So when we talk about a fat being saturated, it means there's no double bonds between any of the carbons. It's attached to as many hydrogens as it possibly can. When it's unsaturated, it's got 1 or more double bonds in there by removing one of the hydrogens. So it's having to share with one of the carbons twice. And when it does that, it's going to cause a kink in the molecule in there. And that's going to mean it's less stable. So if you have a stack of papers that are all nice and flat, and you put them in a pile, that's going to be relatively stable. If there's a little bit of air circulating in the room, that stack of nice, clean, fresh paper is probably going to be okay. However, if you take papers and you bend them all in half and put a couple folds in them, and then you go to stack them up, what's going to happen if there's a little bit of air circulating in that room to all those crooked papers? That pile is going to fall apart more easily. And it's the same thing with the fats. They are going to get damaged more easily when they are unsaturated like that. So that was the reason for making the hydrogenated oils, is if you artificially did that, it would make it more stable, meaning it wouldn't rot as fast and it can live forever like the Crisco in there. Where your normal unsaturated fats, if you have, say you've bought some canola oil and you forgot about it and it sat in your pantry for a couple years, ever found oil in your kitchen like that you forgot that you had, and you open it up and you go, smells kind of weird. It's gone rancid in there. That's when it has spoiled in there. It's interacted with the light and the environment around it, and it's going to have more free radicals in it. It's not good and healthy anymore. So we already talked about the hydrogenated oils and the trans fats in there. So phospholipids, these are going to have 2 fatty acids attached to a glycerol in there. And the third thing is going to be a hydroxyl on there that's joined to the phosphate group. The way we typically draw these is this represents your phosphate end or your phosphate head. And you've got your 2 fatty acids that come down off of there is a real typical way to see those drawn in there. This is going to be in all cell membranes. And the reason that is special is because at this end here, this phosphate end likes water. This end down here does not like water. So if you put another one in there like this, what you have is a surface where you can have it like watery on the inside. It likes watery on the outside, but these 2 wateries don't want to mix because they don't want to have to go through this zone in here. Your sterols, these are going to be lipids that have a ring in them. So they actually have 4 fused rings in there, and the different chemical groups can attach to the rings. The most common one is cholesterol. It's only found in animal cells. Plants don't have cholesterol. They do have other sterols though. and it can contribute to atherosclerosis with your trans and saturated fats. So here is showing your fatty acid attaching to a glycerol here. This is showing an ester linkage that holds it together. So a lot of these things like this, I'm not going to ask you to reproduce any of these chemical structures on here. Right now, it's just so you can look and say, okay, yeah, there's a picture of it, what it looks like, and leave it at that. So this is showing a saturated fat here. An example, butter, lard is another one in there. And that they're all nice and symmetrical. They'll stack up nice and neatly. Where your unsaturated fat, an example, olive oil is monounsaturated. It's got a bend in it. So you can leave butter sitting on the counter and it will be solid. You can put butter in the refrigerator, it's solid. You put olive oil on the counter, it's liquid. You put it in the refrigerator, it's solid. It takes a cooler temperature to make things more stable in there. This is your phospholipid here, so you've got the phosphate head and your two fatty acid tails on there. And then the smaller picture, the simpler one here. So with proteins, proteins are used for structures in the body. They're over 50% of the mass, dry mass of most cells in there. So that's what your body wants to use protein for, is to rebuild structures. It doesn't want to use it as an energy source in there. And the types of proteins we have, enzymatic proteins that will act like enzymes, defensive proteins, they're used to make your immune proteins in there. You can have some that are storage proteins in there that are going to help to store amino acids. So for example, ovalbumin in eggs gets broken down into amino acids to help build an embryo in there. Transport proteins that help move things across cell membranes. Some that act as hormones like insulin, some that will be receptors, some that are going to help with movement like actin and myosin, and some that are just going to make structures like collagen in there. So your building block of a protein is going to be the amino acids, and we've got 20 different amino acids in there. Each polypeptide is going to be made up of one or more of these. So when we look at the structure of an amino acid, you've got a carbon in the center, you've got a hydrogen on one leg, you have this acid group on one leg, an amine group on one leg, and the R group, which is variable, on another. So the only thing that's different on all 20 of them is what you stick at R. The rest of this is all the same all the way through. So they get classified into different groups. So you've got some that are hydrophobic because they've got non-polar side chains. And most of these groups, there's not really a reason to remember who all is in there. You don't need to memorize the names and the abbreviations for them either. In there, but this just shows you some that are in there. The polar side chains, so these are going to be hydrophilic, they're going to like water in there. Some are going to be electrically charged, so some will be acidic, some will be basic. So your groups of them, the ones that are non-polar, the ones that are polar, electrically charged, acid, bases on there. The groups that are more important to remember in biology are these last three here. The essential amino acids are going to be ones that you have to consume through food in there. And the non-essential are ones that your body can make from other amino acids. So years ago, they used to talk about it that a vegetarian would have to do all this food combining in there, And they thought, well, unless they eat these certain foods together, they're not going to get all the different amino acids they need. Now we know you can eat them pretty much at whatever time of the day. We've had vegetarians that didn't do food combining for numerous years, and they're still around. So, nope, you're not going to die from that. But what you do need to have is, at some point, you do need to get all of these taken in there. If you eat animal products, those proteins are going to have all of those different amino acids in there. But say you eat like rice, there would be, say, part of the group in there. Beans would have another portion of the group in there. Corn might have another segment of the group in there. So for a vegetarian, it's just a need to have a variety to get all of the different ones in there. The branched chain amino acids are these three. The significance in those is they're metabolized in the muscle rather than the liver. So people who exercise a lot would want to make sure that they have adequate amounts of the branched chain amino acids in their diet for muscle repair. So amino acids get linked together by a peptide bond. And the peptide bond is kind of important to know. You'll see that over and over again in biology. It's going to be joined when the carboxyl group next to the amine group comes off. They're going to form water, a dehydration in there. So what's going to happen is you would put you're going to take and one of the hydrogens in here to make water. So doesn't really, it's kind of hard to show it in there without taking off the wrong things in there. So protein structures. A functional protein, what you end up with is you can have your functional protein that may be made up of just one polypeptide, or it may be made up of multiple polypeptides in there. But what they do is they fold up into their own unique shape in there. And it seems to be spontaneous that they fold up. Nearly all the functions depend on its ability to recognize that shape and bind to another molecule. It's very specific. It's like a lock and key in there. But you have to have the right key. You may be able to fit the key in the hole, but if it isn't the right key, it's not going to hit all the parts inside. Some of them are going to be more globular in shape. They'll be roughly spherical in there. Some of them are going to be more fibrous shape. They'll make more long fibers in there. So we have different levels of structure in there. Your primary structure of a protein is going to be the sequence of the amino acids. So what order are they in the chain? The secondary is going to be looking at these segments that get either repeatedly coiled or folded. And so you're going to have some that will repeatedly coil into the alpha helices in here. Some that are going to repeatedly fold into these beta pleated sheets. The tertiary structure in there is going to be the overall shape from the interaction of the R groups. So you make these coils like this, and then you fold it up some more in there. So it's kind of like an origami thing where you've got your first folds that you do, and then you do more folds on top of it. And whatever is here is going to start to interact with its neighbors in there. So that's where you're going to get, when you have a lot of cysteine in there, you can have these sulfurs get close to each other, and they make these disulfide bridges, which are those really strong bonds in there. We'll also have hydrophobic interactions where it will make these little cores that don't have any water in there. And then the quaternary structure is going to be the overall structure that results from multiple polypeptides. So that would be like taking your origami things where you make two halves with a piece of paper and then you combine them together with two pieces of paper, making the final structure in there. Except with a protein, you may have multiple polypeptides that end up making your final protein in there. So just some examples of how important it is that these are in the same order. If you have one amino acid different in there, it can affect the shape of a red blood cell in there. So with the normal, they're going to be this biconcave disc that's roughly spherical in there. If you switch out one of those amino acids, it will change the shape of it enough that now it ends up making a sickle-shaped cell. And that's what happens with sickle cell anemia, is it's just one amino acid different in there. Let's see where, okay. We'll finish proteins, and then all you'll have to do is the nucleic acids at home. So denaturation is going to be when a protein loses its shape and it becomes biologically inactive. Sometimes it can be reversed in the lab, but a lot of times in nature it cannot be. So an example would be if you take an egg and you fry it in there, you put in some heat, and you have changed the bonds in there. You can't take that egg and put it back to being a raw egg again after you fried it in there. It's denatured. Temperature is one of the things that can denature things in there, also pH. So one of the concerns, if you're looking at a human and their body temperature gets too high, you don't want your body temperature getting up over 105. At that point, you can start to denature some of the proteins in your body and end up with permanent damage from your temperature being too high. The chaperonins are these chaperone proteins. These are going to help the protein get folded correct, and it also helps keep it away from interfering influences in there so that it doesn't unfold as easily in there. So you can take the same sequence, and if you have the same sequence of amino acids, it always folds exactly the same every single time. So how do we know the 3D shapes of these? X-ray crystallography is how that has been determined in there. They put an X-ray beam through it and look at the shape that way. Also, nuclear magnetic resonance spectroscopy. It doesn't require you form crystals in there. So with bioinformatics, they can actually computerize predict. If you know the amino acid sequence, you can predict the shape. So we're going to stop there today.